So, data fitting. So we saw yesterday, with Virginia's talk, some of the motivations for why you would fit models to data. And we're just going to go over that a bit more. So why do we fit? We fit to data so we can make inferences, conclusions about that data. So we saw that with Virginia. Also tomorrow you'll see us fit models to data for machine learning. But here we are fitting models to data to understand the data better. So rather than trying to do predictions with these models, we are interested in the parameters of the model because it tells us something about the image and something about potentially the biology of what we've fit the model to. Whereas tomorrow you'll see us doing predictions based on models. There's two different ways of using models. And this is sort of the definition that I'm using. Data fitting is the process of fitting models to data and analyzing the accuracy of fit. Engineers and scientists use data fitting techniques, including mathematical equations and non-parametric models, to model the acquired data. So data fitting is quite a broad term, and it includes all of these aspects in it. So here's some data, some simple slightly noisy data, and there's clearly visually a relationship between x and y. But what are x and y in this context? Well, this represents the relationship between two continuous quantitative variables. And we have one variable, the x, which is known by many names because there are many disciplines that focus on these relationships. But the definition I like to use the best is most often is the independent variable, so x is our independent variable. And then we have y, and y is known through other names as well, target, response, outcome, but I like dependent. We're looking to see if y is dependent on x, we call it the dependent variable. And what we want to do is we want to fit a line to this data so that we can understand the relationship between x and y and work out if there is a statistical relationship. And I think that mastering linear regression is an excellent starting point for mastering data analysis. It's the most simple model you can fit to data as far as I'm aware, and using it, we can use it as a test case to explore the different ideas for fitting. So what we want to do is we've got our data, and we want to fit, in this case, a straight line to the data. And this straight line has an equation. So from your maths at school, you probably would have come across the equation y equals mx plus c. When you start doing data science and things like that, you tend to change the notation as well. And here we have y hat, which is the y of our model, is linked to our input, our independent variable x, and b0 and b1. So b1 is equivalent to m, and b0 is equivalent to c in the old notation. We just do it like this because we may have very many independent variables, so we don't want to keep using letters of the alphabet, we use subscripts. But there's no difference between this equation and y equals mx plus c. So b0 is the offset where the line crosses the y-axis, and b1 is the gradient of this line. And when we're doing data fitting, what we want to do is we want to learn the values of b0 and b1, which best allow us to fit this line to this data. Okay. So how do we do it? Well, this is the analytical method. And you can apply analytical methods to a number of models, and I'll explain when you can do that. But what we're basically doing is, rather than trying to play around with the parameters and experiment with them and optimize them, either manually or using a numerical method, we are basically able, with an analytical method, to jump straight to the solution, as in we understand enough about the model that we know exactly how to go straight to that data without having to go through iterations of experimentation when we're fitting it. And we can do that using these equations. So b0 can be calculated by summing all of your points in their y coordinate and subtracting b1 times all of your coordinates summed up in x over the number of coordinates points that you have. But we need to know b1, but b1 can be calculated here by multiplying xi and yi, summing it, subtracting the number of particles times the mean of our x and y, blah, blah, blah. You get the idea. So we can jump straight to the solution through doing that processing of our input data. And this is what we call an analytical solution. And this can be defined as an analytical solution to a problem is one that we have a proof, a series of logical steps that can be followed and verified as correct. So it would be fantastic if all models had an analytical solution, because it means you could jump straight to the best way of fitting that model to data. But the reality is we don't often know enough about the models that we're fitting in terms of how they really how they work mathematically, that we can't have an analytical solution. Okay, this is very good for simple models and models where we put a lot of time to really understand how they function. The alternative, actually, I'm going to go, before I talk about the alternative, I'm going to talk more about how we get to this analytical method in linear regression, and just try and give you a bit of an intuition for it. So what we want to do is, ultimately for our analytical solution, we want to minimize the error between the points and our model. So here, I hope you can see, we've basically put our x and y coordinates into our equation to come up with a series of equations with the same unknowns in each case. So what this green bit represents is this line here. This is the error between our points and our line in the y-axis. And so for the first point, the error is represented by 6 minus b0 in brackets times our x-coordinate b1. So you can see we have a series of equations here that are all giving us clues about what b0 and b1 may be. And then what we want to do is minimize the sum of these, so we can express the sum of these like this. Of course, you can use more compact notation to do it, but I'm just really showing you how all of those unknowns would fit in. And we know that our loss function, so this is the shape of this function here, if we vary b0 and b1, is convex. And what I mean by convex is it basically looks like this. There is one global minima, okay? And typically, if there's one global minima and the solution is convex, then you can jump straight to the solution. It means there is an analytical solution. But this will only happen with simple models, and we'll only be able to prove it for more complicated models if we really understand the mathematics behind it, okay? So as long as we've got enough data points, so we need to have more data points than we have unknowns. This is another prerequisite. As long as we have more points than we have unknowns, then we can perfectly calculate where the global optimum is without having to iterate, okay? So this is the sort of definition of an analytical proof. If you want to know how we know that this is convex, then there's a link here that you can follow. And it basically comes down to the fact that if a loss function has a second derivative and that behaves well, then we know that it will have one solution, and that is convex. So if that's the case, partial derivative, and then what you do is you set them both to 0 and you rearrange them, and what you get is these equations that I already showed you for b0 and b1, and this is basically finding the point at which this curve is 0. Okay, this is the way in which you can get straight there through partial derivatives. So you don't really need to understand, follow that here, but there's plenty of resources online where you can work it out yourself, and it's nice and elegant, and it's a good way of getting into calculus and linear algebra. But as I pointed out, the reality is a lot of models that we may use, for example this one, they're not so mathematically pure. So this looks a bit like a sigmoid, but it's not a sigmoid, a sine curve. It's got weird kinks in it and things like that. It's a complicated model, and we don't understand the mathematics of it perfectly. I think Virginie had really nice examples yesterday of fitting like outlines of flies to to the flies itself. Those models are not, although they define mathematically, we don't really understand them completely in terms of their global minimums and things like that. So in terms of fitting it, we have to do it numerically. We have to use numerical optimization. And so this is sort of the alternate to an analytical method. This is when we don't really fundamentally understand everything in terms of the error function of a particular model. So we can't just jump straight to the solution in terms of the fit. We have to experiment. Okay? And so this is what a lot of numerical optimization is about. It's about altering and iterating over the parameters to find the best parameters to fit that model to a certain data set. So here we've got that data again. We've got our data points. The model will appear in a second. And then you can imagine associated with that there is a certain error distribution where you have parameters. This could be multi-dimensional, not just two, it could be many dimensional. And here again I'm showing a nice sort of convex error function where there is a global minima, but you can imagine this could be more complicated. But what we want to do when we fit our model, so this is just we initialize it, we throw it on there, and we'll have a certain error, for example here, and then we want to play around with it until we find the optimum. You can see now the error has gone down to where the global optimum is. So basically what we want to do for numerical optimization is sort of crunch through the numbers and try and find the best parameters to fit the model. And we can do that in different ways. So here are some optimization algorithms visualized. And the idea is that depending on our error function, which can vary dramatically, and it can have many sort of sub-minima, more than one minima, and there may be one true global minima, or there might be more than one global minima with the same value. They can be very complicated, and there are different optimization algorithms. This is what tweaks those parameters and try and finds the best parameters for the situation. There are many ways in which they can try and approach the problem to do it as efficiently as possible, right? Because you don't want to just do a grid search. You don't want to exhaustively try all of your parameters. You want to engage with your loss function and work out how you are doing when you tweak certain parameters. Is it improving fast? Is it getting worse? The relationship of our model to our data. So there's a whole art to doing this, whereby you apply different optimization algorithms and see how well they optimize your model, or if they fail completely, and also what initialization parameters should you use. So this is a big area of science. You know, there are a lot of mathematicians and statisticians that spend their whole lives just playing around with optimization functions and things like that. Okay, but we typically, in my bit of science anyway, we typically just try and pick an algorithm that works. You know, you try and initialize in a sensible way, and then you try and pick an algorithm that will do the optimization for you. And if it works 99 times out of 100, or if it even works 9 times out of 10, you'll probably use that algorithm. Okay. So, although I want to engage with you and show you that there's lots to be discovered here, to get the benefits of a lot of other people's work, you don't have to understand it yourself completely. My understanding only goes so far, but I continually try and do more and more optimization because I find it fascinating. But don't be put off by the apparent complexity of it. Okay. And in the practical, you'll see some nice worked examples where we can see optimization working in practice. Hopefully it's accessible to you. So two of these sort of libraries that you can use for optimization. There's fundamentally there's this scientific Python library, which allows you to do optimization and root finding. And there's also a library which I like to use all the time called the LMFit library, which is basically a container for this, a wrapper for this, and it also has other functionality for curve fitting. So data fitting basically is inclusive of curve fitting, but curve fitting also has things about smoothing curves and things like that. But if you're seeing words like optimization, data fitting, curve fitting, you're basically in the right department for fitting distributions to image data and signal data. So in the practical, we'll use LMFit. It's very easy to install. And now I'll talk you through a sort of general pipeline that you'll use for fitting data like that. So first of all, you need to prepare your data. You need to format your data in the right way. Maybe do some pre-processing on it, some noise reduction, for example. And then before you fit anything, you need to make sure you've chosen a good model, or defined a good model. In the practical, you see we define models for Gaussians and for lines, and then we try and fit them. And then parameter initialization. And this is something I asked Virginia yesterday, is like, how do you draw that first cell? How do you get that first model onto your image? And that's basically initialization. So you have to think carefully about how to initialize your model, so that when it starts to initialize, so as it starts to optimize, it can make some headway, it can make some sense of what's there. If you just initialize a model in the top left-hand corner of an image, don't expect it to find your cell on the right-hand side. You have to give it clues as to where to start, and it does the sort of very subtle work of getting it to fit perfectly, if you see what I mean. So initialization is very important. Once you initialize, there is then an iterative cycle where you fit the model, you evaluate the error, then you tune the model parameters, and you repeat this. And those algorithms that I showed you before, they all do this in different or subtly different ways, and they will work better on certain problems over others. If you invest more and more time, you'll understand when to use some over others, but as a beginner, you should really just go with the default and then try and understand if it fails, why it's failing, and then try and find a solution to that. It's hard, without context, it's hard to really understand why you should use one over the other, but if you keep doing it and keep learning, then you'll get better at it. And then once your optimization has proceeded either for a certain number of iterations or until the error has reduced to a certain amount or is not changing any further, then this cycle will stop, and then it's up to you to then review your parameters in the context of your model. So generally you take your fit parameters and you put them into the model, and then you visualize them and see how well visually it's fitting the raw data. And yeah, so review and visualize are very similar, and we'll do this in the practical. And this can all be done in a series of lines of code. Okay. And it's nice when it works, it feels solid. I've achieved something. So when you're reviewing the fit, you'll get you have the potential to look at a lot of parameters. I'm not going to go through all of them, but I do want to point out residuals and also chi-squared. So these are good metrics for assessing how well the algorithm is fitting. You want to minimize your chi-squared, and the residuals represent, I think it's actually the next slide, yeah, your residuals represent the difference between your observed values minus the predicted values of your model. So this is basically the error between your model and the data you're trying to fit. And what you want to do is you want to minimize that, but typically what we will do is we will visualize it. So for every point that we have, we measure the error between our model and the X point, and what we're looking for is error which is randomly distributed around 0, so some points will be higher, some points will be lower. The key thing is, if you start seeing patterns in your residuals like this, it implies there's a systematic error in your model with respect to your data, and you've probably chosen the wrong model to try and fit. Okay, so you want your residuals to be random, so it's important to visualize and look at them that way. But the chi-squared is kind of a measure of the total error across all of your residuals, and in theory we want to reduce that, but there are also other metrics that we can use. So you should always try and fit the simplest model, get the English right. You want to fit a simple model, and the simpler the better. Obviously you want it to fit well, which is where the complexity comes in, but if you can get away with a simple model, you want to use the more simple model. And these metrics, AIC and BIC, will tell you, give you a measure of the accuracy of your fit, but also take into account the number of parameters. So you really want to, because the more parameters you have, you will be able to fit the data better. That's just the nature of having more parameters. But you may be overfitting, you may have too many parameters. And so you always want to try and have fewer parameters, because this is a more powerful model, as long as it fits everything that you've got in the way that you want. Okay. So So there's a lot of things to investigate here, but just want to point out those few. So we will continue on to time series. So this is kind of to join up with Stephen in the afternoon. It turns out there's a lot of data fitting in time series data. So what do I mean by time series data? A time series data is a series of points indexed or listed or graphed in time order. Most commonly, a time series is a sequence taken at successive equally spaced points in time. So you're either taking, for example, an image at regular time points, or you're detecting just raw fluorescence from a pinhole or something like that over a period of time series. So, but here are some popular microscopy examples. So there's MSD analysis, where you basically tracking, and then you correlate the signal, assess whether molecules are diffusing or not, or if they have a certain directed motion. There's Storm and Palm, where you use time series to do super resolution imaging, and we'll go into how that works. There's fluorescence correlation spectroscopy, where you look at the temporal correlation of a signal over time, and also fluorescence recovery after photobleaching. This is another popular technique, or it has been popular, where you photobleach a part of your specimen, and then you look at the fluorescence recovering in that region over a series of time points. Okay, so we're going to go into a few of these. So first we'll start with storm imaging. So this is stochastic optical reconstruction microscopy. It's a popular super resolution technique, where what we do is we create an environment with directed storm, at least, where our fluorophore will blink stochastically and sparsely over time, and then will go dark. So we're constantly, we're basically stopping a lot of the fluorescence that you'd usually get from a sample. So it just blinks at sparse locations, and then what we do is we measure. We'll go over this in the subsequent slides, but we measure the locations of the blinks that appear, and then we fit a Gaussian to them. This is where the data fitting comes in, and then we can reconstruct a super resolution representation based on the error that we're measuring with each of these blinking fluorophores. And so with directed storm, what we do is we use a combination of laser illumination and a special dye, which means that the fluorophores will blink at a very sort of limited rate over and then photobleach over the acquisition period, allowing us to see these individual bright spots over time. So here's an example. So imagine there's a sample here which under a normal illumination would appear very bright and it would look like a cell or something. In the paradigm of storm, under these illumination conditions, physical conditions, chemical conditions, then we have a number of blinks on our image, and what we do is we over time record frames and record where these blinks are occurring, and what we want to do is fit these locations, work out exactly where they are, and measure the number of photons that we get in each case. And what we do to do that is we fit a Gaussian, or often known as a normal distribution. And this is a very popular distribution I'm sure all of you have seen at some point. It appears in biology very often due to central limit theorem. So what central limit theorem says is that if you take any distribution and you sample that distribution a number of times independently, then if you repeat that process many times, then the mean and the standard deviation of those samples of the original distribution will appear as Gaussian. So that basically means there's many ways in which, just through physical effects, that a Gaussian distribution will be measured in biology. Encourage you to look up central limit theorem, because it's very interesting, and there are many ways in which this appears in biology. But the Gaussian, for example, is a very useful distribution to know, and we're going to use it here to fit our storm data. So again, because this is a relatively simple distribution, we can try and fit it analytically, but just through the nature of the distribution and the fact we have these lobes at the sides, it doesn't work so well with noisy data. So rather than using the analytical parameters, we just use these to estimate where our Gaussian is, and then we use a numerical technique to actually fit the distribution, and we'll see this in the practical. And it's very easy to generalize the Gaussian to multiple dimensions. So this is a visualization of the 2D Gaussian, which has more parameters. And this is the 3D Gaussian here, which also has additional parameters. Usually in imaging, we have anisotropic Gaussians, so Gaussians which are longer in the z-axis versus the lateral xy axis. And that's why we have different sigmas for the different axes, and that allows us to change the shape of the Gaussian. And it pops up time and time again in imaging. And what we want to do is, I mentioned that we have these sparse detections in our image. What we want to do is fit Gaussians here, and get the coordinates and measure the sigma and also the amplitude of these by fitting. And then we want to reconstruct our image here. Okay? But how do we know how big to do these Gaussians? Because these are diffraction limited, and we want to go beyond diffraction limit. But due to the notion that our Gaussians are actually linked to the number of photons that have been measured, we can actually go beyond the precision of the diffraction limit. So this again relates to central limit theorem. You can treat these photons that have been measured in this exact location as a sample of the mean of the underlying distribution. And the more photons you have, the sharper the distribution ends up being because of this relationship here. This is part of central limit theorem, where the sigma of the resulting distribution of the samples, the sigma equals the sigma of the underlying distribution divided by the number of photons n here. So if you have detections with many photons, you can basically have a high precision for the accuracy of this location than what we saw in the diffraction limited image. Okay, so this is central limit theorem coming up and allowing us to basically break the diffraction limit of light. So the trick is to be able to identify each for independently, which is what we do with the stochastic imaging profile, and then we fit the distribution, and then due to the nature of the signal, we're actually able to have a greater precision in how we reconstruct the image because of our knowledge of the underlying physics and of the statistical process that leads to it. So what we can do is, rather than representing the image like this, which would be our Gaussians with the diffraction limit, we can go to a super resolution representation like this, where the size of the Gaussian is really linked to the precision of our detection. Okay. But obviously we have to fit a lot of Gaussians to do this, and this is where the data fitting comes in. So you'll see in the practical later, we're not going to reconstruct a storm image, but we go through basically all of the steps. We have an image with Gaussian distributions on it, and we try and isolate those, and then we fit Gaussians to them, and we read out the parameters of those Gaussians. And that's basically what you would do in the storm pipeline. The optimization algorithm and the approach that we're using is very robust in the practical. Generally, there's been different approaches which have tried to do the process faster and also with the same level of accuracy. So it's worth reading about storm and harm reconstruction algorithms, but there are many ways of doing it, and they all have their own caveats, and they all compete with each other to say they've got the best algorithm. But ultimately, in my experience, they all have different weaknesses and strengths. But what I'm sort of giving you is the kind of the way in which to access that topic. I'm showing you the sort of robust way, and then from there you can understand some of the caveats of the different approaches. And then fluorescence correlation spectroscopy. This is another form of time series analysis, and this is what We've done a lot in my previous laboratory, and what Falk and Aurelian also do right now a lot. So how do we do FCS? How does it work? Well, we often want to quantify, the reason we do FCS is we often want to quantify the rate of diffusion of proteins in cells or in a synthetic system. But what we generally have to go on is just an acquisition like this, where we're seeing fluctuations over time within our cell, and we know these fluctuations mean something, but we're not sure how to extract the data. This is where FCS comes in. So we want to find out the diffusion rate of these proteins in response to certain treatments, because it relates to biological mechanisms, and that's interesting. So how do we do it? Sorry, there's some more history. It's been around since the 1970s and has evolved several times with different technologies. These days it's most commonly applied using confocal microscopes with specialist detection hardware for single photon counting. So you could define single photon counting as not really being a time series, because we don't acquire the data on a regular time scale, we just detect all of the photons. But FCS can also be done by integrating photons over regular time intervals, which is well within the definition of time series, and that's what I'll show you now. And also these days, stimulated emission depletion, which is another very popular form of super resolution, is combined with FCS, so that we can actually do FCS measurements in a tighter and a restricted PSF. So basically doing FCS within a super resolution regime and being able to measure diffusion in a very specific type volumetric area. But how does the FCS work in general for the default case? Well, we measure our intensity at high temporal frequency in one or more locations. We look at how the intensity in the spot fluctuates with time, and it's this flux of fluorescence particles in and out of the observation volume, the rate at which this happens, which we can measure to calculate the rate of diffusion. And we use correlation analysis to yield the strength and duration of these fluctuations, and I'll show you that. And then once we've got our correlation profile, we can then fit models, which we'll do in the practical, and not just calculate the rate of diffusion, but also estimate the number of fluorescent molecules that were there at any one time. So I'll show you this. So this is the bit you've been waiting for, one of my animations. So what we do in a typical point FCS experiment is we choose a single point on our cell, and then we illuminate just that area with our confocal illumination profile, and then using the pinhole we remove the outer focus light. So we're just left with the emission from molecules that are moving through our detection volume. You see, then over time, molecules will move in and out, and we generate an intensity profile like this, which is our time series. So, anyone want to see my animation again from the beginning? Yeah? So we shine our laser there, and that looks like this. Sorry. Looks like this. And then these are going to start moving, and then we generate our profile like this. Cool, huh? And then once we got our time series signal, we want to analyze the fluctuations in the signal and find out how long the fluctuations last. So how long does it take for a particle to move in and out of the observation volume? We do this through correlation analysis, and specifically autocorrelation in the case of one signal. Oh dear, look at this. This is awful, isn't it? So there is an equation here. Maybe I can adjust. No, I can't do that. Okay. So there is an equation here, which is the autocorrelation function, and you'll see it again in practical, so don't worry so much about it. But what this basically does is it takes one copy of our signal, and then it moves it along temporally in comparison to the initial signal, and then we compare the self-similarity of this signal using this equation here. And what happens is we end up with a curve like this. So at the beginning it's flat. That's because we haven't moved so far in time that things have had time to change within the observation spot. So we've taken so many measurements so quickly that our doing that quicker than the molecule can move. And so it's self-similar at this point. But then as we shift one signal, and remember it's the same signal, but we shift it temporarily, as we move it more and more, then the self-similarity decreases because the molecules are no longer, the same molecule is no longer in the observation spot, so the fluorescence has changed, and over time it will change so much that it no longer recognizes it itself. You get this characteristic curve here, which gives us the average transit time of the molecules. So if the molecules were moving at different speeds, we'd expect this point of inflection to be at different locations along this axis here. So I'll show you the animation again. So rather than looking at the top part, if you look down here, you'll see how the curve generates as we go further along in how we displace this signal here. So this is autocorrelation. We take the same signal, we shift it a little bit, measure the similarity, shift it some more, measure the similarity, and then you get a characteristic curve like this. And then there are certain visual aspects of this which you can use to evaluate it. So if the curve's further to the right, it indicates that whatever you're looking at is diffusing slowly. If it's further to the left-hand side, it means that it's diffusing very quickly, and so changes occurred very quickly in what you were looking at. And then the height of the curve indicates the level of concentration. Inversely to what you might expect, the higher the concentration, the lower the amplitude of the curve, and this is because correlation has a stronger signal if there's a relatively sparse amount of molecules within the observation spot. If the observation spot is very dense, then it's hard to distinguish through this processing when molecules have moved in and left out. Basically your signal to noise is lower because you're really looking to see when changes occur to the overall level of fluorescence. So if it's always molecules coming in and out, then these changes are subtle, and so you get a stronger, higher amplitude if there's relatively few molecules there. Obviously, if you have no molecules there, then it doesn't work. So there's a kind of a balance between having just the right, not too many, but not too few molecules. Okay, so that's kind of how FCS works and how you interpret it. But in reality, what we do is we fit a model. And you'll see in the practical that we do this, and what we're really interested in is the transit time. So this is the point at which the curve is at 50%, and this represents the average time it takes for the molecules to move through that observation spot. And then if we know the size of the observation spot, which we do, we can relate that directly to the rate of diffusion, which is the value, it's the concept that biologists like to use to interpret the rate of movement. So you can, there are certain models that you fit, and then you can calculate diffusion. Again, this appears in your in the practical, but there are many models which can account for different artifacts and different aspects of these curves. For example, you may have multiple diffusion modes there. You may have different species of protein with the same tag. You may have, as I said, different modes where the fluorescent molecules will diffuse differently depending on where they are on the cell or what's happening to them. So there are many models and things that you can apply. And this is a classic case where you should try and apply a model with as few parameters as possible, because it can get very hard to understand what all the parameters are really doing in the context of your biology, and also the robustness of the fit will reduce, be more susceptible to noise if it's a more complicated model. But yeah, so here's some example of the equations. But don't worry, we're not going to go into all of these in the practical. I'm not going to ask you to derive these in the practical. But in general, like any fitting operation, use fewest parameters as possible, which are sufficient for your needs, and check the residuals for systematic failures. So something that can happen, for example, in FCS is photo bleaching, and also there can be problems with the detector. Maybe there's some kind of noise that's been influencing it. Because we're doing this correlation, which really looks for patterns in the signal at high frequency, can be susceptible to systematic problems like that. So check your residuals and make sure there's nothing strange going on there. And then, yeah, just to remind you about the residuals, what they represented again It's the difference between your measured data and your model-based prediction. And this is some software that I've developed in previous years. This is Python-based software where you can do the processing, the correlation, and you can do the fitting of the curves, and you can put in models and stuff like that. I wrote it in Python from scratch, but it uses the LMFit library for the fitting, and it also uses something called PyQt to make the GUI. And then what I did is I compiled the Pythons as a standalone application using a library called PyInstaller, and I then compiled it on 3 different virtual machines, one for Mac, one for PC, and one for Linux, so people could run it with a double click wherever they were on whatever computer without having to worry about installing Python and things like that. Don't try and do that. It worked very well. People become very fond of the software who use it, but it's a nightmare to try and compile things across multiple operating systems. But what I found is certain people don't want to install Python things themselves. They don't want to see the code. They just want to use the application. And by making an executable which is independent of what Python you've installed, you can do that, but it's quite an undertaking. So only for the fearless. These days I sort of recommend using Anaconda and making sure your libraries are very easy to install on something like Anaconda, because most people can handle installing Anaconda and just running a few commands to get your software running. And then you can still have a GUI and things pop up, but they've got this kind of environment with the necessary libraries. But just so you know, you can compile Python code into a standalone application. And there are things out there that you might not realize have been written in Python because they've done it so well and they've compiled it, but it is painful. It's not really your job as a scientist to do that, but that's how you can. It's a rabbit hole which you can go down. You might have noticed I go down quite a few rabbit holes. Okay, so that brings the presentation to an end. I might have mentioned my Twitter account earlier but now we're going to go into the practical. It's the same format for my practicals yesterday. If you have questions, then we'll be walking around, and we'll keep doing this practical until 12:30. Again, a lot of you won't finish it, so don't worry about that. Just try and engage with it and understand all of the different aspects as you go along. And yeah, you can always email me questions about it and things like that if you don't finish. Any questions? No? So thank you very much.